Welcome everyone to our session on experimental design for chemistry at O levels. In this presentation, we'll be covering the essential skills needed to conduct scientific experiments and analyze data effectively, which are crucial for success in your O-level chemistry exams. The hands-on experience gained through well-designed experiments will not only help you understand chemistry concepts better, but also develop critical thinking and problem-solving abilities. As you can see in the image, we have various laboratory equipment like beakers, test tubes, and scientific diagrams on the chalkboard, which are all important tools for conducting chemistry experiments. Throughout this session, we'll explore best practices for experimental design, data collection, and analysis, equipping you with the knowledge and confidence to excel in your O-level chemistry assessments. Let's dive in and get started. Experiments are the foundation of the scientific method. They allow us to systematically investigate research questions. Identifying and isolating the relevant variables is crucial for obtaining reliable, meaningful results. Carefully planning data collection procedures ensures the integrity and accuracy of our experimental findings. This slide introduces the key elements of experimental design that will be covered in more detail throughout the presentation. The scientific method is a structured approach to understanding the world through observation, experimentation, and analysis. It starts with observing a problem or phenomenon, then forming a hypothesis to explain it. Next, experiments are designed and conducted to test the hypothesis. The results are carefully analyzed to draw conclusions that support or refute the original hypothesis. Finally, the findings are communicated to the broader scientific community. This cyclical process allows scientists to continually expand our knowledge and understanding of the natural world. The key to a successful experiment is developing a clear, testable hypothesis. Carefully consider the problem you're trying to solve and make an informed, logical guess about the relationship between variables. Your hypothesis should be specific, concise, and based on existing knowledge or preliminary data. Be sure to document your hypothesis clearly and precisely in your lab notebook. This will guide your experiment design and keep you focused on testing your prediction. The image on this slide shows various scientific lab equipment like beakers, test tubes, and pipettes set up on a wooden table with a chalkboard background. This slide is focused on identifying the different types of variables in a scientific experiment. The first key variable is the independent variable. This is the variable that the researcher manipulates or changes to observe its effects. The second key variable is the dependent variable. This is the variable that is measured or observed to see how it responds to changes in the independent variable. The third key variable is the controlled variables. These are all the other factors that must be kept constant in order to isolate the relationship between the independent and dependent variables. Understanding these different types of variables is crucial for designing valid and reliable scientific experiments. Experimental variables are the key components that researchers manipulate and measure in a study. There are four main types of experimental variables. Independent variables, the factors that the researcher changes or controls to observe their effect. Dependent variables, the outcomes or responses that are measured to determine the impact of the independent variables. Controlled variables, factors that are kept constant to isolate the relationship between the independent and dependent variables. Confounding variables, external factors that may influence the relationship between the independent and dependent variables, introducing bias. Controlled experiments are the foundation of the scientific method. The key steps are: define variables, identify all the factors that could affect the outcome. Isolate variables, manipulate one variable at a time while keeping all others constant. Measure outcomes, observe and record the effects of the manipulated variable. Careful experimental design is crucial to ensure the validity of results. By isolating variables and measuring their effects, researchers can determine causal relationships and draw reliable conclusions. The slide introduces the concept of experimental controls, which are essential for ensuring the validity and reliability of scientific experiments. Positive controls are samples that should produce the expected result, validating the overall experiment. Negative controls are samples that should not produce the desired result, confirming the test is working as intended. Blank controls contain all the components except the variable being tested, ensuring there is no background interference that could impact the results. These different types of controls help researchers identify and account for any confounding factors, isolating the effect of the variable being studied. Understanding and properly implementing these controls is crucial for drawing accurate conclusions from experimental data. Randomization is crucial to ensure unbiased results and eliminate potential confounding factors. Conducting repeated trials helps account for natural variability and increases the reliability and statistical significance of the findings. Randomized controlled trials are the gold standard in experimental design, using both randomization and replication to isolate the effects of the independent variable. These principles of randomization and replication are fundamental to robust, high-quality experimental research. Safety in the chemistry lab is critical to protect ourselves and others. First, it's important to wear the proper attire, a lab coat, safety goggles, and closed-toed shoes to guard against spills and splashes. Loose clothing can get caught in equipment, so we want to avoid that. When handling chemicals, carefully read the labels and follow the instructions for safe handling and disposal. Use the appropriate personal protective equipment, like gloves or a face mask, when working with hazardous substances. Ensure the lab is well ventilated to prevent the buildup of fumes. Use a fume hood when working with volatile or toxic chemicals. Finally, know the location of important safety equipment like showers, eye wash stations, and fire extinguishers. Review emergency protocols so you're ready to respond quickly if an accident occurs. Equipment and materials are essential for conducting chemistry experiments at the O-level. Key glassware includes beakers, test tubes, and Erlenmeyer flasks. Important tools are tongs, spatulas, and pipettes. Common materials are acids, bases, indicators, and various chemical compounds. Proper safety gear like goggles, gloves, and lab coats is crucial. Access to balances, hot plates, and other lab apparatus is needed to carry out experiments effectively. Precise measurements are critical in the lab. Use calibrated instruments like graduated cylinders, micrometers, and analytical balances to ensure accurate and reproducible results. Maintaining tight temperature control is key. Use thermometers, heating/cooling devices, and temperature monitoring to minimize sources of error. Timing is also crucial. Utilize stopwatches, timers, and clocks to precisely record the duration of reactions, heating/cooling periods, and other time-dependent processes. Accurately measuring liquid volumes is essential for procedures like titrations and dilutions. Employ pipettes, burettes, and other calibrated glassware to get precise volume measurements. Accurately recording data is crucial for the integrity and reliability of your research. Proper labeling of data points, measurements, and samples ensures everything is clearly identified and organized. Using standardized units consistently throughout the experiment allows for accurate comparisons and analysis. Detailed qualitative observations provide important context to supplement the quantitative data. Handling samples and equipment carefully prevents errors or damage that could compromise the data. Effective data visualization is key for interpreting and communicating experimental results. Graphing data allows patterns, trends, and relationships to become clear. Common graph types include line plots, scatter plots, bar graphs, and histograms. The choice of graph depends on the type of data and the insights you want to uncover. The image on this slide shows an example of a colorful line graph representing scientific data trends. The data analysis phase is critical to extracting meaningful insights from the experimental results. We'll carefully examine both the quantitative and qualitative data, looking for patterns, trends, and any anomalies that could provide valuable clues. Determining statistical significance is key to ensuring the conclusions we draw are valid and not simply due to chance. We'll employ appropriate statistical methods to make this assessment. Finally, we'll formulate logical conclusions that directly address the original hypothesis and research question. It's important we don't overstate or overreach beyond what the data actually supports. Titrations are a common technique for determining the concentration of an unknown solution. The process involves adding a standardized reagent until a color change or other indicator signals the endpoint. This allows you to calculate the concentration of the unknown solution based on the amount of reagent added. Distillation is a technique for separating a mixture into its component parts. It works by taking advantage of differences in boiling points, allowing you to purify substances. Chromatography is a powerful separation technique that can identify and quantify the individual components of a mixture. It works by passing the mixture through a stationary phase, which causes the components to separate. Spectroscopy analyzes how matter interacts with electromagnetic radiation. This can provide information about the structure, composition, and properties of substances. Calorimetry measures the heat energy released or absorbed during a chemical reaction or physical process. This allows you to determine important thermodynamic properties, like enthalpy and entropy. Titrations allow us to precisely measure the volume of a titrant solution needed to neutralize an analyte solution. The setup involves using a burette to carefully add the titrant until the endpoint is reached, indicated by a color change. Acid-base indicators like phenolphthalein change color at the neutralization point, signaling the equivalence point. Accurate volume measurements and close monitoring are critical to get reliable results that can quantify the concentration of the unknown analyte. Distillation is a powerful separation technique that allows us to purify liquids by taking advantage of differences in their boiling points. The process involves heating the liquid mixture, causing the more volatile components to vaporize first. These vapors are then cooled and condensed back into a pure liquid. Distillation has a wide range of applications, from chemistry labs and industrial processes to the production of alcohol and essential oils. It's an essential tool for isolating and purifying substances, with the ability to achieve high levels of purity. Chromatography is a powerful analytical technique used to separate and identify the components of a complex mixture. It works by having a sample mixture move through a stationary phase, with the help of a mobile phase, allowing the different components to separate. Common chromatographic methods include column, paper, and thin-layer chromatography, each with unique applications and capabilities. The image shows a typical setup for column chromatography, with a glass column filled with a solid adsorbent material and the sample mixture separating into distinct colored bands as it travels through. Chromatography is an essential tool in many fields, from chemistry and biochemistry to environmental analysis and forensics, allowing researchers to isolate and study the individual components of complex samples. Spectroscopy is the study of how substances interact with different forms of electromagnetic radiation. There are several key types of spectroscopy, each providing unique insights. Absorption spectroscopy measures the wavelengths of light absorbed by a substance, revealing information about its molecular structure and chemical composition. Emission spectroscopy analyzes the wavelengths of light emitted when a substance is excited, providing insights into its atomic and molecular properties. Infrared spectroscopy studies the absorption of infrared radiation, which can identify functional groups and provide structural information about a substance. Each of these spectroscopic techniques offers valuable analytical capabilities for understanding the properties and composition of materials. Calorimetry is the measurement of heat transfer during chemical or physical processes. It helps determine the enthalpy changes associated with reactions, phase transitions, and other thermal events. Calorimetry experiments use insulated containers, thermometers, and precise measurements to capture the heat flow. Calorimetry is used in chemistry, biology, and materials science to study energy changes. Effective time management is crucial in experimental design. Carefully plan the sequence of steps and allocate sufficient time for each procedure. Consider the availability of equipment, materials, and lab space when designing your experiment. Adapt your design to work within the given constraints. Determine the appropriate sample size to ensure statistical significance. Plan for data analysis techniques that will allow you to draw reliable conclusions from your results. Identify potential sources of error in your experiment and implement strategies to minimize them, such as calibrating instruments and following precise protocols. Time management is critical for success in the lab. It helps you stay organized, focused, and productive. The key is to prioritize your tasks, identify the most important and time-sensitive experiments, and focus on completing those first. Create a detailed timeline for your experimental procedures, accounting for setup, data collection, and analysis. This will help you stay on track. Minimize distractions as much as possible. Eliminate unnecessary interruptions and stay focused on the task at hand to maximize your productivity. By implementing these time management strategies, you'll be able to get more done in less time and deliver high-quality results. Limited funding can constrain the equipment, materials, and facilities available for experiments. Experiments must be designed to fit within the class time or lab schedule constraints. Access to specific chemicals, reagents, or other required materials may impact what experiments can be done. The capabilities and restrictions of the available lab space and equipment need to be taken into account when designing experiments. Determining the appropriate sample size is crucial for ensuring the statistical significance of your experimental results. Key factors to consider include the size of the population, the expected effect size, and the acceptable margin of error. Applying the right statistical techniques, such as t-tests, ANOVA, or regression analysis, can help you draw reliable conclusions from your data and identify any patterns or relationships. Carefully analyzing the results, including measures of central tendency and dispersion, can provide valuable insights into the significance and implications of your findings. Experimental errors can come from many sources, and it's crucial to be aware of them to improve the quality of your research. Measurement errors can arise from inaccuracies in equipment, improper techniques, or environmental factors. This can skew your results. Sampling errors occur when the samples you study don't accurately represent the larger population. This can lead to unreliable data. Uncontrolled variables that you fail to identify and control can introduce confounding factors that compromise the integrity of your experiment. Human errors made by the researcher during setup, data collection, or analysis can also undermine the validity of your findings. Being vigilant about these common sources of error is crucial for ensuring the reliability and reproducibility of your experimental results. Systematic errors are inherent flaws or biases in your experimental design or equipment. To minimize these, you should calibrate instruments, control variables, and repeat measurements. Random errors are the natural variation that occurs in measurements. To reduce random errors, you should increase sample size, perform multiple trials, and use statistical techniques like calculating standard deviation to quantify and account for the variation. By addressing both systematic and random errors, you can improve the accuracy and reliability of your experimental results. The structure of a lab report is crucial for effectively communicating experimental results. Key components include organizing the report into clear sections. Data should be presented visually through graphs, tables, and charts to help the audience understand the findings. Thorough analysis and interpretation of the data is necessary to draw logical conclusions from the experiment. A well-structured lab report allows the audience to follow the flow of the experiment and grasp the significance of the results. The structure of a lab report is crucial. It should have clear sections like introduction, methods, results, and discussion. Formatting is important too. Use consistent headings, citations, and other formatting to make the report easy to follow. Thoroughly document your process, observations, and data analysis. This provides important context and transparency. Present your findings objectively and avoid bias or speculation. The goal is to communicate your work clearly and accurately. The data we've collected reveals some interesting patterns and trends that provide valuable insights into our experiment. Based on our analysis, the results support our original hypothesis and demonstrate the significance of our findings. However, we should also acknowledge some limitations in our experimental design that may have influenced the outcomes. Moving forward, there are a few areas we could improve to strengthen the quality of our research, such as adjusting our methodology or expanding the scope of our study. These ideas for future improvements could help us build on what we've learned and uncover even more meaningful insights. The key takeaway here is the importance of well-designed experiments in chemistry. Properly planned experiments are critical for obtaining reliable data and drawing accurate conclusions. As you continue your chemistry studies and research, focus on continuously improving your experimental design process. Apply the lessons you've learned to refine your approach and become an even more skilled researcher. Beyond what we've covered, there are many advanced experimental techniques to explore. Seek out opportunities to gain more hands-on lab experience and continue expanding your knowledge and skills in this area. Experimental design is crucial for conducting rigorous, scientific research. A well-designed experiment ensures a systematic process to test hypotheses and draw reliable conclusions. Proper experimental design helps minimize bias and errors, providing high-quality, trustworthy data to support research findings. A thoughtful, optimized experimental design can save time and resources by efficiently using materials, equipment, and time. This allows researchers to conduct their work in a more cost-effective manner. Iterative improvement. We've learned a lot from our current experiments, and we can use those lessons to refine and optimize our experimental design for future investigations. By continually improving the process, we can get better results. Leveraging automation. There are many opportunities to automate data collection, analysis, and reporting. This will improve efficiency and reduce the risk of human error in our work. Interdisciplinary collaboration. Bringing in experts from related fields can give us new perspectives and innovative approaches to incorporate into our experimental design. Diverse expertise will strengthen our work. Continuous learning. It's crucial that we stay up to date with the latest advancements in experimental design methodologies and technologies. This will ensure our practices remain cutting-edge and effective. This slide introduces the key resources available for learning more about experimental design. It covers 4 main areas: online courses, textbooks, scientific journals, and expert guidance. The online courses provide interactive learning on the fundamentals of experimental design and data analysis. The textbooks offer in-depth coverage of design principles, statistical methods, and real-world case studies. The scientific journals showcase the latest research and best practices in the field of experimental design. And expert guidance, through researchers or workshops, can provide personalized advice and feedback on your specific experimental designs. These resources can help deepen your understanding of experimental design and improve the quality of your research.